And so our next speaker is Lisa Zheng, who worked with Mr. Zachary Wissner-Gross at MIT, and she will be speaking on an inhibitory model for neuronal symmetry breaking.
Thank you, Dr. Rickert. Before I begin talking about my project, I'd like to give you guys this basic background on neurons. So, in the initial stages of growth, a neuron is a cell body with a bunch of intermediate processes called neurites growing out of the cell body. Over time, as this neuron grows, these neurites compete with each other, and eventually one neurite will grow to become the axon, while the rest of them remain short and become the dendrites. Now, this neurite growth is a very complex process, and this neurite competition. There are two types of mechanisms proposed as to how this neurite competition goes about. The first type of mechanism is called growth proteins. There are two types of growth proteins, activators and inhibitors. Activators are proteins that help these neurites grow, while inhibitors are proteins that slow down the growth of neurites. The second type of mechanism is called inhibitory signals. Inhibitory signals are signals sent from the tip of each neurite to the other neurites. The longer the neurite, the stronger the inhibitory signal. Now, as I said before, this neurite competition is a very complex process. So there have been two types of models proposed to help try to understand this process. The two types are depletion and inhibition. Depletion models are models that only take into account growth proteins. They have a certain concentration of growth protein in the cell body, and over time, this growth protein gets shipped out to the tip of each neurite. The second type of model is called inhibition. Inhibitory models take into account both the growth protein and the inhibitory signals. As these neurites are growing, growth protein gets shipped out to the ends of these neurites, while inhibitory signals are sent from each neurite tip to the other neurite tips. Now, there have been many depletion models proposed so far. The one that I worked with was a very classic model proposed by Samuels using these four equations. Ti represents the transport. C naught represents the concentration of growth protein in the cell body. Ci represents the concentration of growth protein in each tip of the neurite I, while Li represents the length of each neurite I. As there have been many depletion models proposed to date, but so far there have not been any inhibition models proposed. My project was proposing the very first inhibition model. Working with my mentor, we modified the Samuels model and added on this chi-C term. Chi-C we added multiplied by the summation of this protein concentration of every cell tip of each neurite tip, subtracting the protein concentration in neurite I. The reason for this is the inhibitory signal is based on the concentration of all of the other neurites except for that neurite. We proposed two different kinds of inhibition models, a concentration model and a length-based model. For the length-based model, we added a nearly identical term except based on the length and using a chi-L term. What we did with our models was we compared them to the results already found in the Samuels model to see how our inhibition term would affect. So the first result that we found was concentration-based inhibition will accelerate axon formation. When chi-C equals 0 is the regular Samuels model. But we found that as you increase chi-C, the rate of axon formation increases. We found this holds to be true for both concentration-based inhibition and length-based inhibition. The second result that we found was in the Samuels model, he had a graph on which, on the x-axis, he graphed chi-1, which is a diffusion term, and on the y-axis, he graphed chi-2, which is a positive feedback term. He varied the values of chi-1 and chi-2, and he drew this line, which the range above is the range of axon formation, while underneath is the range of no axon formation. We found that as you increase the value of chi-C, the range of axon formation increases. We found this holds to be true for both concentration-based inhibition and length-based inhibition. The third result that we found was in the Samuels model, he had two graphs. One in which he cut, at a certain time point, he cut the longest neurite to a certain length relative to the second longest neurite. When he cut it to just above the length of the second longest neurite, the cut neurite would regenerate to become the axon again. However, if he cut the longest neurite to a length just under the second longest neurite, it would not regenerate to become the axon. What we did was we graphed cutting a neurite to a certain percentage relative to the second longest neurite, and then we graphed the probability, the fraction of the time that it would regenerate to become the axon. We found that as you increase the value of chi-C, the inhibition term, shorter cut or axotomized neurites would regenerate for the concentration-based inhibition. However, interestingly, when we use the length-based inhibition model, it seemed to have no effect on cutting this on the cutting of the neurites. We hypothesized that this was because when you cut a neurite, you're affecting the length, but you're not affecting the concentration, meaning that the concentration will still allow the neurite to regrow out, while the length will not. Along with our numerical analysis, we also did some analytical work in which we solved the Samuels equations and we solved our inhibition models analytically. We found that in the steady state approximation, when time goes to infinity and all of the factors have been held stable, this C0 term will go to a 1 over chi2. And for there to be axon formation, chi2 must be greater than the number of neurites. We found that this parameter holds for both the Samuels model and for our inhibition models. Now, this parameter for axon formation is different than the graphs that I showed you earlier, but we hypothesized that the reason for this is because inhibition modifies the rate of axon formation, not the stability. In other words, the inhibition modifies how fast axons form, not whether or not they will form. So in conclusion, my project, we proposed the first concentration and length-based inhibition models. We found that for both concentration and length-based inhibition, axon formation was accelerated, and there are a larger range of parameters over which axons will form. But we found that for only length-based concentration, will there be shorter cut neurites are allowed to regenerate. Also, that chi2 must be greater than n for there to be axon formation. I would like to acknowledge my mentor, Zach Wissner-Gross, for helping me with this project, my tutor, Dr. Jenny Sendova, as well as the RSI staff, CEE, MIT, and my sponsors, the Department of Defense, and Building and Engineering and Science Talent. Thank you.
Are there questions? Yeah, Professor Holt.
Yeah, could you go back to your first model slide?
This one?
Yes, yes. So you introduced two terms, both a length-based term and a concentration-based inhibitory term, and those are both linear. Is there any reason why you chose a linear term and a concentration?
So the question was, why did I choose to have a linear-based term? The reason was we based this off of the Samuels model, and Samuels himself chose to have a linear-based model. So going along with his choice of having a linear-based model, we just simply modified his and kept along with the linear-based model.
Are there other questions? Mr. Carr?
Is there experimental work that you did that goes along to corroborate the results of the model that helps you select how or assess how accurate I-go-to-2 work is making?
So the question was, did we do any experimental work to verify how accurate these were? No, we did not. But in our further work, we are planning to potentially do an experimental work in which we could design a project to verify these results.
So I guess a follow-on question to that is, can you put this in context for me of, so you took an existing model, you formulated some other alternate models, but what do I take away from that without any way to assess whether you described to me the behavior of those two models, but I don't have any way to compare reality in the models?
So the question is whether or not, like, what he can take away from this since we don't have any experimental work. Well, what we did was we mainly did computational-based work. So doing some computational-based work gives a good background for doing an experimental work. We do plan to possibly do some experimental work in the future to help verify these results.
Can I ask you about your method for solving these simultaneous equations? In other words, how did you do it? What algorithm did you implement?
Okay, so the question was the method for solving these equations. What we did was we took these equations and we put them in the steady state, so that in the steady state, what happens is the C naught term, the dc dt term, the C naught term goes into 1 over chi2, and we set that in the steady state, these dc dt, dci dt, and dli dt go to 0. So using those, we were able to solve the equations and substitute in the equations and solve to see that chi2 must be greater than n for there to be axon formation, and that C naught goes to 1 over chi2.
Time-dependent solutions, how did you do this? So those are the asymptotic solutions, but what about the time-dependent solutions?
Can you clarify what you mean by time-dependent solutions?
Your answer to the question previous told me about the asymptotic solutions, i.e. when d on dt is 0.
Yes.
What about the time dependence? I'm asking actually about the numerical algorithm that you used to solve the equation.
Okay. So he asked about the numerical algorithm that we used to solve this equation for time-dependent. We actually did not do any time-dependent. All of the solving that we did was all in the steady state.